Okay, so we're going to carry on today with muscles. So the learning goals for muscles: name the 3 types of muscle and describe their locations in the body. Explain the differences between the muscle's origin and insertion. Identify and describe skeletal, cardiac, and smooth muscle, and that's on a cellular level as well as their overall function. Identify their general locations and purposes, and know the difference of flexion and extension and which muscles in the body oppose each other. So just to recap, we have 4 types of tissue in the body overall. So you have epithelial tissue, which includes hair, skin, nails. We have all epithelial tissue on the inside of our body. It lines all of our cavities, so it would line the uterus, the vaginal canal, the mouth, the abdomen, and the thorax. Nervous tissue includes gray matter in the brain, the spinal cord, and nerves. Connective tissue, we talked about fat, and that's produced in the yellow marrow of the long bones. We've got fat, fascia, which we'll talk about today, ligaments, and tendons. And then the last type of tissue in the body is muscle. And we break that down into skeletal, cardiac, and smooth muscle. And smooth muscle can be broken down into multi-unit smooth muscle and visceral smooth muscle. Okay, so these are the 3 cellular types and general muscle types in the body. We'll talk about each. We'll start with skeletal muscle, which is also known as voluntary striated muscle. So skeletal muscle is controlled by the conscious mind and moves the bones of the skeleton. It's connected into insertion points of the bones of the skeleton, all those processes that we talked about in class previously. So it's called voluntary, meaning that we are able to control the actions of skeletal muscle. However, some of the muscles in the skeleton go on in cruise control, so they sort of go onto autopilot. Think about the muscles of the diaphragm and the external and internal intercostal muscles. They all relate to your breathing. So when you're asleep, you're not able to actively tell yourself, breathe in, breathe out. Instead, your body clicks into cruise control and it maintains your breathing throughout. A few different locations for skeletal muscle. We've got head and neck, cutaneous, abdominal muscles, thoracic limb, and the pelvic limb, and muscles of respiration. So the muscles of respiration I just mentioned, the 3 of them, the external and internal intercostal muscles. So those are the muscles within the ribs, between the ribs. And then you have your diaphragm, which is the big dome muscle at the base of your thorax. Cutaneous muscles are kind of unique. We don't have cutaneous muscles in the same way that horses and cats and dogs do. They have that ability to twitch their skin to get rid of flies or mosquitoes that land on them. And that twitching of the skin is called paniculus reflex. This is the great drawing of a cell. Just to recap what the nucleus is all about, that was the main goal. And the nucleus stores the DNA. So looking at our skeletal muscle cell, this is what it looks like when we break it down. So we have this is the muscle itself. Okay, the widest part of the muscle is called the belly of the muscle. And then within the muscle we have various tubules, these cylinders, that contain numerous skeletal cells. So these are skeletal cells, also called skeletal fibers or muscle fibers. Muscle fibers is what they're most often called. And just to note the pattern, that it's one large cylinder, which is the muscle, that contains smaller cylinders, which contain muscle cells in general. So the muscle fibers, also known as muscle cells, they're extremely long, so they're completely different than any other cell in the body. A normal cell in the body would be 0.001 millimeters in length or diameter. A muscle cell, so a muscle fiber, can be numerous inches long, which is excessively long for a cell. They have numerous nuclei, so nuclei is the plural of nucleus. So those are all the nuclei that contain genetic material, and they can have lots and lots of nuclei. They're arranged in a cylindrical pattern, And within the cylinder of the muscle cell, they have all these other cylinders called myofibrils. And the myofibrils are a certain arrangement of protein within the cell. And it's because of the myofibrils and their arrangement under the microscope that we call it striated. So voluntary striated muscle means that it looks striped when it's under the microscope, and that's because of the arrangement of the myofibrils. And just to note, as with most cells, but in specific with skeletal muscle cells, one muscle cell, so one little muscle fiber, would never work on its own. It's working with all of these muscle fibers to create a movement of the muscle overall. Next, we have cardiac muscle, which is also called involuntary striated. So thinking about the cellular components, you can already guess that it has myofibrils because of its striated appearance under the microscope. So cardiac cells are only found in the heart. You'll find them nowhere else in the body. And they have something interesting about them in regard to efficiency and energy utilization. So if you flex your hand really hard and hold it for about 30 seconds, you'll notice that your muscles in your arm and your hand will start to fatigue, and then they'll stop working, and your hand will release. Cardiac muscles are quite different than skeletal muscles in that they never stop working until you die, of course, or the animal dies. But they'll continuously work, so they don't fatigue, they don't have a period of rest, they're always working. So some interesting points about them. They have these intercalated discs that other muscle fibers don't have. So intercalated discs, which is where they get their neural activity from. So the neuron impulse is received there, and they create the contraction. They do have myofibrils, like we talked about, so their arrangement with these little protein, the arrangement of their protein within the cell. They generally only have up to 2 nuclei, and they're located within the center of the cell. So this is one cell. Okay, it stops there. This is one cell. This is another cell, and it's a cross section. And the intercalated discs allow the energy to move between each cell. So those neural synapses to move in between each cell. So it has 1 to 2 nuclei per cell. And they're in a branch. Rather than being a nice long strand like your skeletal muscle, they're branched. Their actual appearance is in a branch. Okay, last we have we have smooth, also called non-striated involuntary. So smooth muscle we break into 2 different types. We have visceral smooth muscle, and we have multi-unit smooth muscle. Visceral smooth muscle is contained in large sheets of cells in the walls of hollow organs. Multi-unit smooth muscle are small, discrete groups of cells, and they work on their own. So the visceral smooth muscle, you could find it in the uterus, in the bladder, anywhere in the gut, so the stomach, the intestines, the colon, and it's responsible for large wave-like motions. So if you think about when an animal urinates, their bladder is contracting in one wave-like motion to push the urine out of their bladder. Likewise with peristalsis in the gut, it's a wave-like motion that's pushing that little bolus of food down the GI tract out into the colon. As opposed to your multi-unit smooth muscle, which works in tiny little contractions, very fine movements. Visceral smooth muscle does not need a stimuli, so it does not need an external stimuli. It responds to stretching of that hollow organ. Multi-unit smooth muscle does require a stimuli in order to work. So an example of multi-unit smooth muscle would be the ciliary muscle, ciliary body, within the eye that allows one to see things close up and see things a little bit further away. Other example of multi-unit smooth muscle, you'd find it in the respiratory tract, you'd find it in blood vessels, and that's more or less it, and within the eyes. Okay, this is the breakdown of the of the visceral smooth muscle, or smooth muscle in general, both multi-unit and visceral. And just to note, it's a very simple cell. It's a spindle-shaped cell, has one nucleus, and it does not have myofibrils. Rather, it has all its nuclear content scattered within the cell itself. So this is a chart that is probably a good chart to know that identifies the differences, the physical characteristics of each of the three types of muscle cells. Okay, just getting into a little bit of anatomy of the muscle and the name game. The name game gets really significant with muscles. Myo generally refers to muscles. Sarco, which we'll talk about in CLP 400, sarco specifically refers to muscle cells. So just going back a little bit, when I talked about the 4 different types of tissues, so we've talked about muscles, and now we're going to talk about connective tissue. So connective tissue, a few different types of connective tissue. We have fat, which the white fat within this animal, you can see it throughout the organs, okay, cushioning the organs, cushioning the abdomen. And the benefit of fat is it acts as insulation for the animal. It also stores some vitamins. And then we have fascia, which is a smooth, thin layer of connective tissue that is over all the muscles in the body. So when you have a piece of chicken, so a chicken breast in front of you, and you pinch the chicken breast, you can pull up a really thin membrane. That's the fascia. And on here you have thinner and thicker layers of fascia throughout this musculature. And then we have ligaments and tendons. So it's really important to note that tendons attach the muscle to the bone and ligaments attach bone to bone. So a great example of a ligament would be the ligaments within the knee, so within the stifle joint. And there are numerous ligaments. There's the cranial cruciate ligament, which is the absolute main ligament that most dogs rupture. And then you have the caudal cruciate ligament, and you have medial and lateral collateral ligaments as well. So numerous ligaments within, especially within synovial joints. And then you have tendons, and a good example of a tendon would be the gastrocnemius and superficial digital flexor tendon, which in people we tend to call the Achilles tendon or Achilles heel. So this tendon connects the gastrocnemius muscle, which is the main muscle for flexion of the hind leg, and it's attached at the distal aspect of the femur, the muscle, and then the muscle turns into a tendon, and it connects to the hock. Okay, the superficial digital flexor, which are really important for movement of the foot, attach to the tibia, work their way around the hock, and connect down through a tendon at the toes. And in dogs and cats, you can get a tear of the digital, this tendon in particular, Achilles tendon if you want to call it. You can get a partial tear where the gastrocnemius tendon only is released. Okay, that way the superficial tendon can still flex the toes, but the gastrocnemius tendon can't flex the foot with the hock. And then you have a full tear where the foot and the hock lay flat to the ground. Okay, tendons and aponeuroses. So aponeuroses are broad sheets of connective tissue, thicker than more broad than a tendon. So it's more like a sheet rather than one thin strand. And the most common, or the one that you'll see the most, the aponeuroses, is the linea alba. So the linea alba is literally a white line once you cut away the skin and fat in the abdomen. It's a white line of connective tissue that reaches from the xiphoid cartilage down to the pubic symphysis of the pelvis. And that linea alba is where abdominal incisions would be made during a spay or during any sort of abdominal surgery. And the surgeons will cut along the linea alba because that allows for the best healing of those muscles after surgery. the linea alba is the area where the rectus abdominis muscles connect to each other. So those are the six-pack muscles. So they naturally lie in that formation, and when an incision's made and sutured back up, they have the best chance of healing through that area. When we're looking at the movement of muscle, it's good to know that they often work in opposition. So when one muscle is flexing, the other is relaxed. And a great example of this, when our biceps brachii, so our biceps in our arm, are flexing, the triceps are relaxed. So the muscle that's flexing is called the agonist, and the muscle that's relaxing is the antagonist. And you can see the tendon attachment to the bone for each. Okay, likewise, when our triceps are contracting, our biceps are relaxing. Okay, looking at here, just to note so that's the true connection. So this is your gastrocnemius muscle, which for animals and people, it'd be our calf muscle. And then there's a very long tendon from the gastrocnemius that connects to the hock of the animal. So the origin of the gastrocnemius is the femur, the insertion is the hock. When we talk about origin insertion, so origin is the term given to the portion of the muscle that stays the most stable during the muscle contraction. So looking at this image again, the origin is on the femur. When this muscle contracts, it's going to tilt the heel up. So tilt the hock up to flex, to point the toes. So the origin remains stable. The point of insertion is moving with that flexion of the hock. Likewise, we have the origin insertion of the triceps. Okay, so the origin is on the scapula, and the insertion is on the olecranon. So if you imagine when your triceps flex, your elbow moves, your olecranon moves. So the insertion point tends to be less stable than the origin. So these are some names to know for each. The prime mover is a muscle or muscle group that directly produces a desired movement. Antagonist is a muscle or group that directly opposes the action of a prime mover. They can't act as both. So biceps brachii, so that's your biceps on the arm, can be a prime mover, but can also be an antagonist when the triceps brachii are trying to flex. So you cannot flex both your biceps and your triceps. A synergist is a muscle or group that contracts at the same time as the prime mover to assist in carrying out its desired action. So the example for this is to flex your hand into a fist. The deep flexor muscles of the distal front limb are the prime mover to flex the hand. However, the superficial flexor muscles are assisting in furthering the movement. Okay, and lastly, the fixator muscle stabilizes joints to allow other muscles to move. So extend your fingers, flex them into a fist, and feel the muscles on the underside of your forearm contract to flex your fingers. The muscles on your forearm, so the muscles on the underside of your forearm are those are your digital extensor muscles, and they're actually flexing those fingers. They're pulling those fingers into a fist. The muscles on the upper side of your forearm are fixed in place and contracted in order to stabilize the wrist. And then just to note, muscles have their names for different reasons. So you have a biceps muscle in the hind limb, and you have a biceps muscle in the forelimb. Biceps brachii in the forelimb is our typical biceps that we're thinking about. Biceps femoris is the large muscle, hamstring muscle that sits on the caudal aspect of the femur. It's lateral aspect. So they're named for different reasons. They can be named for their shapes. So deltoid means triangle. Therefore, the triangle-shaped muscle of the shoulders are called deltoids. Their location, An example would be triceps brachii in the upper arm. Upper arm generally refers to brachii. There's a direction that they can go in, so rectus means straight, transverse is a different different straight direction, and oblique is on an angle. And then the number of heads or divisions. So the terms cephal, C-E-P-H-A-L, refers to head. So from that is derived the term sep. So if we think about bicep brachii, we're noting that it has 4 heads. Biceps brachii has 2 heads. Quadriceps has 4 heads. And that's our horse muscle. That is good to know. And then these are just some videos. I would like you to note the ones on peristalsis, because that's something that we talked about. And that's it.